No matter what age, race, or gender, every human body is formed of tens of trillions of cells, organized in a fascinating system which we only partially understand. Funded by the NIH, the Human Biomolecular Atlas Project, or HubMap, is a multidisciplinary research effort by over 200 experts in more than 50 institutions. Its goal? To create a map of the human body at single-cell resolution, a feat never accomplished before.
HubMap's a really exciting project. The challenges are dealing with data from a large number of tissue centers, many data formats, many assay types, and bringing those together, and then giving people tools in a way that non-specialists can actually do medicine and biology against this really unique, valuable data set.
One of the major challenges is the integration. I think there's a whole slew of disparate data sets and data types that are coming in. That all needs to be integrated. It needs to be registered and mapped, so to speak, to a organ system framework, whether that's a common coordinate framework or a physiological or biological-based framework.
To develop a map of the human body, NIH needs to coordinate efforts for a diverse international team. Challenges include heterogeneous data, remote communication, large-scale data transfer, and the standardization of workflows. All of this has to happen across a dynamic technological, social, and political landscape.
To put together a whole piece of product that we can deliver to the community and make them understandable to the general public while everything is somewhere in the middle of changing is very difficult.
At this point, we have 18 funded groups that we have to try to pull everybody together, get everyone coordinated and on the same path towards similar goals. Each group has a superpower.
Capturing cells in spatial, three-dimensional detail will help us bring together knowledge distributed across multiple disciplines.
If you go to a doctor, they look at you with clinical systems at the body level. If you talk to scientists, they look at the cellular level and they can explain you very complicated phenomena, but not necessarily are able to bring it all the way up to the medical level information. So what drove me was the idea that we need to bring this somehow together in a multi-scale, multi-dimensional database or tool.
My personal hope is that the work that we do in HubMap can then catalyze this as a general approach in many different areas of science. My ambition is that the legacy of HubMap in 10 or 20 years' time is really thinking about and realizing that using a single-cell analysis type approach for tissues, for understanding complex environments is the default way in which science is done.
So we're building a map on healthy human tissue, but ultimately this is going to be, we think, a model for what could be used for a human disease. What does it look like when things go wrong? So to have a nice, if you will, reference map that is incredibly high detailed on a healthy person, it's really going to help us understand what happens when things go wrong, when disease appears.
The need for a common language between several highly specialized disciplines prompts HubMap to become a catalyst for innovation and collaboration. Our need for knowledge has prompted many notable scientific advancements in the past. So our HubMap members are part of a great tradition, and they are only getting started.
Today I'll be giving a walkthrough of the Common Coordinate Framework Exploration User Interface that has been integrated in the HubMap data portal. The CCF EUI searches the search API available via HubMap to find all the tissues that have been placed via the registration user interface for the Common Coordinate Framework. And what this does is tissues uploaded via the HubMap ingestion portal can be placed spatially inside of reference organs, reference kidneys, and reference spleens in the first data release. In the exploration user interface, we can now explore the tissues that have been placed using a variety of methods. On the left, you see the partonomy, which takes us from the body into different reference organs and then pieces of that organ. You can click the right kidney to filter the results by that kidney, or the left kidney, or the spleen in the first data release. Further data releases will be adding new reference organs and more fine-grained control over querying via the partonomy. In the middle, you see the body UI. This shows our reference organs in a reference body with spatially placed tissues inside. On the right are all the tissues that have been placed in the registration user interface. There are currently 116 samples. You can also, in addition to narrowing the tissues down by our partonomy, you can also use the clinical metadata associated with these tissues, for instance, to filter by sex, age, or BMI, and also down to the tissue mapping centers, the ones who are providing tissues. Right now, for data release 1, Florida and Vanderbilt are the only ones with registered tissues via the registration user interface. So we can now filter our searches, and you see the tissues have been updated. You can click on a reference organ to zoom into that specific organ and see the tissues available in it, and then click on individual tissues below. You can shift click or control click to select multiple, and if you look up, you'll see the ones in blue are the ones that you've selected. If you need more space to see the results, you can expand via the full screen button, and you can also make more room via these panels. In addition, if you would like a dark theme, you can switch on the fly between light and dark theme depending on your needs. Further, at the top you can see that there's some help screen. There's a button which takes us to the portal, but at some point may be a bulk download option. And refresh just resets the view that you see. We can also get back to the portal at any time by clicking the HubMap icon. Finally, we can look at the individual tissues. Some tissues we can view in the exploration user interface if they've provided TIFF images, images of the tissues in different ways. As you can see, this one, you can change the color coding of these by adjusting brightness and transparency. Let's go to one with a little bit cleaner interface here. Okay, so here's a tissue. You can zoom in and out, and this is driven by the Vitesse image viewer, also developed for HubMap. You can adjust colors on the fly, and you can change color schemes on the fly. And in addition, you can get to the metadata associated with the tissue on the right by expanding the About pane here. Some tissues do not have TIFF images associated with them, in which case there are two scenarios. And let's reset here. for codecs and 10x data, we can view the visualization on the HubMap portal. As you can see, this also uses Vitesse, but displays the data in a different way, and each of these visualizations will be different depending on the type of data that's associated with it. So codecs and 10x have these custom interfaces like this to allow you to see the data in a visualization. But for the others which don't have a direct visualization at this point, we simply take you to the page for that specific sample. And there are further videos to explain how to use the HubMap data portal, but this just links out to those in that case. The same thing is true for these download buttons. We allow you to just simply go to the data portal for downloading additional data beyond what is provided in the exploration user interface. And with that, that is the current functionality that's available for data release 1 of the HubMap data portal. So, thank you.
Alright, hello everybody and welcome to this little demo video / tutorial about the CCF registration user interface version 0.5.0. My name's Andy, I'm one of the developers of the system, and I'm really glad to show you how to use it properly. So, as you know, it's accessible via this web link. You're going to find it in the description of this video. As you load the page, make sure to click this little full screen button, alternatively press F11. Here we go, we're now in full screen mode. This is the so-called check-in page. This is what you see when you first load the application. So as you can see here, I'm going to type in my first name. My full name is Andreas. And then I go over here to last name. I type in my last name, and there we go. Now, as you can see, currently only the kidney is supported for the registration UI. In the future, we're planning to also support the heart, the lung, and the spleen. So I'm going to choose the kidney here, and I click Next. So this is the registration interface proper. You can see that we have a human kidney here on the left. You can also see that we have on the inside two-dimensional the inner structures of the kidney. Here on the right side we have a representation of a tissue slice that we want to register. Here on the right we have the tissue sample size in millimeters. So for the sake of this example, I'm going to resize that to 20 by 20 by, let's say, 1. Here we go. Now our sample here is resized. Let me actually change the width to 12. Here we go. So, as you can see, if I drag and drop the tissue slice, I can move it around my scene. You can also see that there is a grid overlaid over the kidney, and then as I move, watch what happens to this coordinate text here. As I move my sliver onto the grid, we can see the current position of the sliver in millimeters as well. Another thing I can do is I can toggle to a so-called side view. So then I see the kidney from the side, and I see the Y and Z dimensions here. And then I can position my sliver from this side as well. So let's just say it was taken from here. Let's say the kidney was cut in half. Then we have this tissue slice here. Now I can go back over here, maybe reposition it slightly. Such that it is over here. There we go. All right. So that's the front side toggle button. You're probably going to be using this quite a bit. if you ever find yourself in a situation where you have to resize any of the position or sizing or rotation that you did, you can just click those reset buttons here. Now actually, while we're talking about it, let's go ahead and rotate our sliver around the center mass point, like so. All right. And then once you're done with your positioning, you can go ahead and open this 3D preview here. So just click the 3D preview button, and then you can actually, by dragging your left mouse button, you can move a 3-dimensional camera around the kidney, so that you have a better 3-dimensional impression with depth cues where exactly your slice is positioned. You can close this view by clicking the 3D preview button again. So far for this step, which is the actual registration. Now we come to, we're going to get our data out of the application. So click on next here, and now we can review our data. So we have my first name, last name. We have the current date. That is the current date of the current alignment. We generated an alignment ID for you automatically. This is automatically generated every time you register a new slice. A reference organ ID. This is currently just a static string. In future versions, this is going to be probably dynamically linked to a database, but this is to be determined. And then we have the data we generated. We have the tissue position on the grid in millimeters, the tissue rotation that we typed in, as well as the tissue size, also in millimeters. And then I can go ahead and copy this to the clipboard. And this is important. I can copy the clipboard, and then this window opens, and then you have to hit Control-C on a Windows machine, or Apple button and C on a Mac to actually copy the string, and then you can paste it into any application you desire. Click OK, and then this new button shows up called Register New Tissue. So when I click this, I'm being taken back to the first screen where my data is still in there. So we still want to register kidney, right? Then your old values are still saved. So if you register a lot of similar slices, you can do that rather easily here. So in this case, let's just be simple and just change the size, maybe something like this. Also change the rotation a tad. Good enough. Here we go, and you click next. And then we have a new alignment ID. We have the current date and time. Name is still the same, and here we have the new values. And we can copy the clipboard again, and then copy this string manually and paste it into any application your heart desires. All right. You can also go back between screens by clicking this back button, and then continue by clicking the next button. In the top right corner, you're going to find a little info screen about the current version of this interface with a bunch of links with more information. All right, thank you so much. This was a very short introduction into this current prototype. If you have any questions, concerns, or comments, feel free to reach out to me. Thank you very much.
The goal of the Human Biomolecular Atlas program, or HubMap, is to develop a framework for comprehensively mapping the healthy human body at single-cell resolution. Given that each human body is unique, this is an enormous challenge.
In order to integrate data from many individual bodies, HubMap will create a common coordinate framework, also called a CCF. A CCF is similar to a postal address system in that it defines the spatial positions of major anatomical structures and cell types within a 3D reference body. This HubMap address system is then used to map future tissue samples so they can be explored in their appropriate spatial and semantic contexts. The CCF must account for human diversity. Here I have 3D printouts of the left and the right kidney of the visible human female. As you can see, the number and shape and size of the individual anatomical structures is rather different, even for one individual.
Diverse methods have been proposed to design a human body atlas. Commonly, reference organs such as this one from the Allen Brain Atlas are designed and used to register new tissue into a 3D Cartesian coordinate system. To create our atlas, we first collect information on key anatomical structures and cell types, along with the biomarkers used to identify these cell types. The resulting anatomical structures and cell types plus biomarkers tables, or ASCT+B, are then used to design 3D reference objects that capture major anatomical structures and the general shape of cells in healthy human adults. Data from the National Library of Medicine's Visible Human project is used to create a 3D reference object library. Cross-sectional cryosection, magnetic resonance imaging, computed tomography, and anatomical images for male and female were used to reconstruct several organs. The 3D nested structure for the kidney is shown here. All objects are named and registered within the HubMap Atlas reference system. The CCF can be used to spatially register new tissue blocks. Commonly, the 3D size, position, and rotation of a tissue block is captured by taking photos of the tissue dissection process. However, it is difficult to compute the exact spatial location, rotation, or size of extracted tissue blocks from these photos. The registration software's user interface, or RUI, which is shown here, was designed for usage by experts who collect human tissue and need to document the tissue extraction site. It requires about 5 minutes of training time and 2 minutes for each tissue registration. Currently, the RUI supports gross anatomical tissue registration. Once biomolecular data becomes available, it will be extended to support placement based on biomolecular markers and patterns. The exploration software's user interface, or EUI, makes it possible to explore 2D and 3D tissue samples semantically and spatially across multiple scales. Tissue position data generated by the RUI is used to position tissue blocks. Cell segmentation algorithm results support cell position and cell type exploration. Semantic and spatial search, browsing, filtering, and details on demand are supported. Filters support the selection of sex, age, BMI, etc., making it possible to explore change as the human body grows older or increases in BMI.
The key feature here is we're tying a spatial reference system to an ontological reference system so that we can tie into other things which were also tied to that ontology. Papers, people, cells, research, all kinds of things.
The HubMap Atlas and its functionality will evolve over time and become more comprehensive of the many organs and cells that make up our human bodies. Are you interested in contributing to this historic effort to create an open, global atlas of the human body at the cellular level? If so, please contact MCIU, that is, us, to contribute.
Hi. I would encourage everyone to get their samples from patients that have been open consented. If you do that, your data will go out to everyone on the planet. In contrast, if you get your samples from patients that have not been open consented, then all your data and all your hard work will get put into a restricted database, and its use will be much more limited. So as much as possible, try and get your samples, again, from open consented patients so they can go out to everybody in the world.
Our tissue mapping center at Vanderbilt University is called the Biomolecular Multimodal Imaging Center, and it's a collaboration that includes Vanderbilt University, Vanderbilt University Medical Center, and Delft University of Technology. And the mission of our center is to develop a pipeline of integrated technologies that brings together imaging mass spectrometry with various forms of microscopy and traditional multiomics, with the goal of constructing comprehensive three-dimensional molecular atlases of human tissues. The focus of our center currently is on the human kidney. And we get those tissues either as discarded donor tissues or as tissue blocks from surgical resections. Once we have the tissue blocks, they're assessed by expert pathologists to determine their normalcy. And once they've passed those quality control checks, they can then go on to the full multimodal three-dimensional characterization pipeline. Now, one of the important things and unique things about HubMap is the focus on developing three-dimensional molecular atlases at cellular resolution. Now, for our technologies, to generate three-dimensional images, we have to take serial sections through an entire tissue block. So what we end up with are taking sections, sometimes hundreds of sections, and each of those sections is mounted onto a glass slide. So each of those samples are then run using different aspects of our characterization pipeline, some being analyzed by imaging mass spectrometry, others being analyzed by different forms of microscopy, and then we combine all of that at the end. Specifically, we're generating molecular images of human tissues using matrix-assisted laser desorption ionization, or MALDI, imaging mass spectrometry, autofluorescence microscopy, classical stain microscopy, and multiplexed immunofluorescence microscopy. We collect the autofluorescence on every tissue section prior to analysis with the other modalities. And the reason we do that is so that the autofluorescence can act as an anchor from one tissue section to another. So we have a common modality that spans all tissue sections. What this allows us to do is spatially co-register everything using the autofluorescence, and ultimately allows us to link one modality to another in three-dimensional space. For the samples that will be analyzed by imaging mass spectrometry, first we collect autofluorescence microscopy. We then define specific regions on the tissue to collect various forms of MALDI imaging mass spectrometry data. And once that data has been collected, the nice thing about MALDI imaging is that the tissue remains intact. So following that analysis, we can then perform stains and collect classical histological images from the same tissue sections. For the samples that will be analyzed by multiplexed immunofluorescence, again, first we collect autofluorescence microscopy, and then following the autofluorescence, we then can perform the antibody-based staining and do that in a cyclical fashion to build up the number of markers for specific cell types within the tissue. Once all of the imaging data has been collected, as I said, we use the autofluorescence data to first register each of the modalities within a single tissue section. So for instance, registering the autofluorescence to the imaging mass spectrometry to the PAS stain microscopy, and create a co-register dataset for an individual tissue section. We can also use the autofluorescence to construct our three-dimensional blocks and then project each of our individual imaging modalities onto that three-dimensional scaffold, allowing us to ultimately produce high spatial resolution, molecular three-dimensional images of human tissues, in this case, of human kidney tissue. As is the case with all HubMap projects, this is a very multidisciplinary effort that requires the creation and integration of many unique capabilities, including procurement and management of human specimens, development of analytical imaging pipelines, and ultimately the construction of bioinformatics workflows that allow us to visualize the three-dimensional molecular atlases that we construct, and also analyze these large-scale multi-omics data sets. We believe that the data generated by HubMap will provide a new paradigm of understanding for the normal state of organs that will span vast molecular and spatial scales, ultimately allowing us to make new connections between molecular profiles and cell types within human tissues.
All right, so I would like to thank everybody contributing material to this video. So my question to you is, given all of the challenges of HubMap, what do you think is your group's greatest contribution to HubMap so far?
So we've done a lot in the first year and a quarter of HubMap. I think our greatest accomplishments are that we've built what we call the ingest portal, which is what the tissue mapping centers use to contribute data. And that has now been used. We actually have data now, and that's a tremendous accomplishment. It's been a long time coming. And more data is coming in almost daily now that it has begun.
So with all that data coming in for all of these different research groups and tissue mapping centers, how does HubMap address the challenge of integrating such a diversity of data types?
Different groups in the biological front are generating different types of data. And when I say different types of data, they might actually measure the same thing, but using different technologies. So the pipelines that we currently support are basically divided into different modalities and include right now pipelines for the analysis of single cell expression data. So these are the levels of genes within single cell ataxic data. So this is the accessibility of DNA in single cell in different organs or tissues. We have pipelines to support spatial information. So spatial is the location of specific proteins and where they are expressed within a cell and between cells in specific tissues. And the last thing we worked on is image microscopy data and connecting that to something called a mass spec, which can measure levels of metabolites or proteins. So all of these data types are currently supported by pipelines we developed for HubMap. And as I said, no matter what platform or technology you use to generate that data, the outcome will look the same for all of these modalities.
So if I wanted to contribute data to HubMap, or if I just wanted to explore data from donors and organs that have already been registered, how would I go about getting started with that?
Once you log into the ingest portal, you see that there are two parts. On the left is the HubMap ID system. This is where research groups and other data providers go to first register a donor or tissues extracted from an organ. On the right is the HubMap data ingest, and this is where data providers upload the data associated with those tissues. Now let's enter the HubMap ID system. Here, data providers can create a new entity, either a new donor, that is an individual whose tissue has been donated for research, or a new tissue block sample, that is a tissue sample that can be sliced into sections for preservation and analysis. We'll click on donor. Here is where data providers would enter the information. So for instance, there's a place for protocols.io DOI. This details exactly how the tissue was extracted. And there's a place to enter in any metadata associated with that donor as well. This is where an ID for the donor would be generated. We can also search through donors and samples that were already registered, and our searches can be filtered by research group, donor, and organ type. If a tissue is one extracted from the left or right kidney, it can be registered using the Registration User Interface, or RUI. Then the data is ready for submission. Next, let's look at the data ingest. Here is where researchers can upload data associated with a tissue sample. This creates a landing zone in Globus, a research data management service that provides high-speed, secure file transfer and sharing. Depending on the type of data, there can be specific requirements about what data is able to be uploaded and what naming schemas to use, etc. The data then goes through a review process, from new to processing, sometimes to invalid, but eventually data is published. After data is registered, uploaded, and processed, it becomes available via the HubMap data portal shown here. If we select donors, we arrive at a faceted search interface where we can limit our search by research group or creator. Here we can see all the data that this particular creator uploaded. Then you can click on a particular donor. This allows us to search either by the samples or by the data sets that were derived from that donor. Choosing samples updates the search interface to show the tissue samples derived from that donor. And if we look at an individual sample, It will have an interface similar to what we've seen for individual donors. We can now also look at data sets. Let's choose one from the University of Florida. For some data that's been uploaded, there are visualizations. This is one, a UMAP plot from a salmon RNA-seq output. The HubMap portal lets you download individual files so you can run your own analyses. But you can also use the search interface and the basic visualizations that are available without having to download anything. Now, finally, I would like to show the CCF user interface, which has been integrated into the portal. You can access it via the CCF link on the main menu page. This version of the CCF UI uses the HubMap Search API, which lets you search for tissue blocks that have a RUI location. You can then explore those tissue blocks in their spatial context together with additional data, including sex, age, and BMI, all uploaded via the ingest portal.
So now, moving beyond the nuts and bolts of how it operates, what would you consider one of the most unique features of the HubMap data portal overall?
I think one of the unique features of the HubMap data portal is the tight integration of the curation process with the data analytics, the pipelines that are used to process uploaded data sets that come from the tissue mapping centers in the portal, all the way to the visualizations that can be displayed directly in the web browser, which makes it really easy for an end user to start interacting with HubMap data. There's no need to install tools to download any data. You open your browser, you type in the URL, or you pick a data set, and you can start interacting with it immediately. I want to make sure that people will come and say, HubMap is a gold standard for how projects at this scale of this complexity should be set up. We want to make tools that people want to use.
I think HubMap is unique in that it's bringing together medical experts and data science experts in a way that I have not seen before. We're looking at all tissues in the body at a cellular and subcellular resolution and bringing together what are really state-of-the-art approaches in handling data, handling pipelines, handling computation, and making it very easy to use single portal to get at all of this.
So my first question for you would be to describe your role in HubMap.
My role in HubMap is to lead one of the mapping centers. I think one of the things that's exciting about HubMap is we're going to be generating so much data from so many different types of tissues. There'll be data from the heart, there'll be data from the spleen, there'll be data from the lung. And it won't even just be one type of data. There'll be imaging data, there'll be sequencing data, there'll be an extraordinary diversity of data sets that we're going to have access to. On one hand, it's wonderful to have so much diversity in the data that we're looking at. On the other hand, as biologists, we want ways to be able to interpret and reference and compare these different data to each other. And one of the best ways to be able to do that is to have some sort of reference framework, to be able to have a language or an ontology or a vocabulary that we can use to describe these different types of data. And that's really the goal of the mapping centers that are part of Hive. I lead one of them, and Kati Borner at Indiana leads the other. And what we're really trying to do is we're trying to build dictionaries or vocabularies or references that we can use to describe all the data that's eventually going to be generated as part of HubMap.
My question is, what is Sora? Like, what does it do? And then where does the name come from?
Yeah. So Seurat is a software toolkit. It's a piece of software that's been developed by my lab. And actually, it started when I was a postdoc. The very first pieces of code I developed when I was a postdoc in Aviv Regev's lab. Aviv is also a member of HubMap. But now it's a software package that I maintain in collaboration with the PhD students, postdocs, and staff in my lab. And the purpose of Seurat is to provide a piece of software that researchers, whether it's from our own lab or hopefully from any lab, can take single-cell data and interpret their data with this computational program. So when you generate a single-cell data set, there are many questions that you might have. You might wonder if the cells fall into different clusters. You might ask which individual genes or which individual sequences each cell contains. There are a variety of common questions which are really unifying to the single-cell field. And those are questions that we hope that researchers can help answer by using Seurat. So really, Seurat is an R toolkit that helps users to explore, analyze, and interpret their single-cell data.
Great, thank you. So I have another question more about focused on the users. So who exactly is using Seurat and why?
So Seurat is very widely used across the community. We see about 25,000 people download the software every month. And we hope it's a very wide variety of users who are able to access the software. Those would include PhD students and postdocs in academic labs, but also researchers in industry and government labs as well. We've tried to make the software accessible to a very wide group of people. So that would include people who have a predominantly computational background, who are very interested in using advanced analytical algorithms. But we also aim to have the software to be accessible and used by people who are just starting out in single-cell analysis and who may not have had a lot of computational experience before, but still need to have a way to be able to analyze and explore their data. So we hope to be able to reach a wide user base because all of those groups of people are generating single-cell data and are interested in the insights that can be derived from looking at it.
Thanks so much. So I want to zoom out a little bit for the next question. And I would like to know, what exactly is data harmonization and why is it necessary?
We've previously discussed some of the big challenges for HubMap, and one of the big challenges is taking many, many different and diverse data sets and interpreting them together. So I'll give you an example of why data harmonization is so important. Let's say that you're doing an experiment on the human heart, and you're looking at cells in the human heart. There are many different types of technologies that you can run to profile those cells. For example, you can use a sequencing technology, for example, single-cell RNA-seq. And what that technology does is it asks what DNA or RNA sequences are present in each cell at any one time. So it's really a molecular profile, a very high-resolution molecular profiling of that cell. On the other hand, you might also use an imaging approach. You might use a microscope, or you might use a spatial profiling technology that allows you to take a picture of that cell and understand where it's actually located. If you use that sort of approach, you might get much less information. You might not know as much about the genes that are present in that particular cell, but you'll know something more about its spatial location and its shape and its morphology. So these are very, very different types of data, right? But fundamentally, they must be linked somehow because they're both being generated from the heart. So we don't want to be able to interpret them completely on their own. We want to be able to understand how they fit together. And if we can understand how they fit together, then we can understand how the molecular content of a cell also influences its spatial location and its morphology and its function. So the goal of data harmonization is being able to take multiple data sets which share some common underlying features and being able to interpret them in a single downstream analysis. And that is really one of the most exciting and difficult challenges for HubMap.
Thank you. And since you just mentioned HubMap, so how has Seurat been used for HubMap or for the HubMap portal so far?
So there are two particular areas in which we're excited about deploying Seurat both now and in the future of HubMap. The first is a complex problem which we call cell annotation. So we have many, many different types of cells in the body, of course, and we'd like to be able to give them a name, right? We'd like to be able to say this is a neuron, or this is a skin cell, or this is a fibroblast, or this is a T cell, or of course this could be a different subset of T cells or a subset of macrophages. We'd like to be able to name cells. And importantly, we want to be able to do that in a consistent and uniform way across all of the HubMap data sets that we generate. We don't want to have individual names for individual data sets. We want to be able to use a consistent and common naming framework. And that's a process that we call cell annotation. And you can imagine that in order to be able to have consistent and uniform names across many different data sets, that is fundamentally a problem for data harmonization. And so that's one of the key areas that we're excited about, is being able to annotate and name cells in a consistent way across HubMap. A second and a very related problem is how can we visualize all of these data together in one common space? So many of you, if you're watching these videos, will be familiar hopefully with the HubMap portal, which is a way that you can visualize, explore, and interact with HubMap data. As I mentioned, there'll be hundreds and eventually thousands of experiments that are generated as part of HubMap. And what we'd like to be able to do is to enable you to visualize and interact with those experiments in a harmonized way, not necessarily having to click on 1,000 individual links and look at 1,000 individual analyses, but to be able to, example, pool all the data sets together from an organ to be able to look at a single integrated and harmonized visualization and downstream analysis. So that's another feature that's enabled by our software and which we hope will be very useful for HubMap going forward.
Thanks so much, Rahul. And that brings me to the last question, which is, how can I, just as a single user, use Seurat?
So Seurat is a fully open source software package. So all of our code and documentation are freely available for you to access. You can go to my lab website, satijalab.org, or just search for Seurat in Google, and hopefully you'll be able to find it. And you'll find a few things. The first is a link to our GitHub page, which contains all of our code and also instructions to install the software on your personal computer or server. But we also have a series of documentations, tutorials, and vignettes, which are basically ways that we think we can help you get started with analyses, particularly if you haven't worked with analyzing single-cell data before. They're sort of guided analyses where we walk you through a data set that's in the literature that can kind of illustrate the types of commands and analyses that you might want to run on your own data when performing a single-cell analysis. So we hope that all of that documentation and vignettes can help a new user kind of familiarize themselves both with Seurat, but also with single-cell analysis in general.
We all have memories of seeing our first real library, of encountering endless shelves of books and wondering how anyone could find what they might be looking for in such a large collection. How would you know if the library has anything in your area of interest anyway? Well, every book has a code. It might be a Library of Congress code based on a classification system created in 1897. It might be a code from the Dewey Decimal System, created even earlier in the 19th century. We classify books on the basis of their content, and we use these classifications to find new information, to put books back in the correct place, and to assess a library's holdings. Classifications pervade our everyday life. Just like the Library of Congress, Amazon classifies its books to help us find publications we might be interested in, and, more important to Amazon, to bring our attention to books it thinks we might be persuaded to buy. All e-commerce sites categorize whatever it is they want us to buy. eBay has an online product catalog that is different from Amazon's. Craigslist organizes its content using an entirely different system. Each site on the web recognizes that its survival depends on how it categorizes its contents, and how well the terms that it uses match the way consumers think about the products that are being offered. It's not much different in biomedicine. Biologists may not be trying to sell anything, but they often need to categorize the things they study and to group things together to clarify their common properties. We call this categorization an ontology. Ontologies are often visualized using an indented tree structure, as in a file browser. This ontology of amino acids, developed at the University of Manchester, tells us that alanine, along with asparagine, cysteine, and glycine, are some of the aliphatic amino acids. A biologist can examine this ontology to know what amino acids exist, what their properties are, and how one might classify them. An ontology may exist only as a conceptual scheme in our heads. We all categorize and classify the things in our world, but we seldom are explicit about writing down our classification systems. When we do write down our ontologies, they become available both to people and to computers. If we want to know what kind of amino acid alanine is, or if we want to write a book about alanine, or if we want to state that we performed an experiment to measure the concentration of alanine, then an ontology can give us a set of standardized terms and relationships that can facilitate all of these things. Our ontology can tell us that aromatic amino acids are not aliphatic, and that amino acids have other properties, such as a standard genetic code. Biomedicine is simply replete with ontologies. Nearly 300 years ago, Carl Linnaeus proposed his binomial nomenclature for speciation. Linnaeus's hierarchical structure of categories of life forms, with each class of organism having a unique genus and species, was a seminal accomplishment. It's still with us today, and perhaps the best-known scientific classification. In the 19th century, the precursor to what is now the International Classification of Diseases, or ICD, appeared on the scene. The ICD provides an ontology of diseases that the World Health Organization uses for international public health activities and that the United States uses for all medical billing. By the turn of the 21st century, a huge international effort was underway to develop an ontology to represent the properties of gene products, the biological processes to which they contribute, the specific molecular function that they perform, and the cellular component with which they are associated. The result was the gene ontology, perhaps the most widely used ontology in bioinformatics. The gene ontology is instrumental for interpreting many high-throughput experiments, and it provides a comprehensive, well-curated digital knowledge base of basic biology. So ontologies are everywhere in biomedicine, standardizing the way that we refer to basic biological processes, to human diseases, and to everything in between. The whole idea of ontology is credited to Aristotle, who formalized the notion of categorizing what exists in the world. Ontology became the branch of metaphysics concerned with the study of existence. In recent years, however, the study of ontology has leapt from the realm of philosophers to that of computer scientists, who create data structures to encode what entities exist in some application area, what the properties of those entities are, and how the entities relate to one another. The desire of biologists to use computers to catalog the results of gene expression is not far from that of the software engineers at Amazon who refine the company's product listings. We are all engaged in work to enumerate what exists in our respective domains, to clarify and communicate properties and relations. We do this to create a description of what exists in some particular application area for the benefit of people who seek clarity regarding the structure of some human experience, for software agents that may need to process information about some application areas, and perhaps most important, for the interaction between people and computers, so that people can be explicit about what they mean, and so that computers can act on human input in the most precise manner possible. We create ontologies as standards, so that we can have agreed upon terms for describing the things that matter to us. There is no need to redefine speciation, for example, when Linnaeus already solved that problem. Having such standards allows interoperation. I can compare my data to your data if we call things by the same name. Computers can also reason about things reliably and efficiently when we use standardized terms and IDs to name things in the world. There's another side effect that occurs when we develop ontologies to describe the entities in an application area that matters to us. We solidify scientific communities when we all use the same language to refer to the same phenomena. Science advances when we can agree on what exactly we are studying and what we should call things. The HubMap project is a large undertaking supported by the United States National Institutes of Health that relies heavily on biomedical ontologies. The goal of HubMap is to create an atlas that can describe every type of cell in the human body, and to describe the characteristics of cell types in terms of genomic, proteomic, and other biomarkers. HubMap needs to be precise in how its atlas talks about the anatomical location of cells. It needs to be precise in how its atlas talks about cell types and biomarkers. There is no other way to obtain the necessary precision than to use standardized ontologies. When scientists who are working on HubMap think about collecting data about an organ such as the kidney, they recognize that the anatomy of the kidney includes a set of substructures called calyces that are all wrapped in an outside layer called the renal cortex. Each of the calyces has constituent parts that are understood all the way down to the cellular level. The HubMap investigators are capturing information about the kidney and about many other organs in the body in a format that lays out the anatomical substructures, the constituent cell types, and for each cell type, the known biomarkers that identify cells of that type in the particular anatomical location. These tables for anatomic structures, cell types, and biomarkers are known as the ASCT plus B tables, and they depend heavily on ontologies. The anatomical structures need to be represented explicitly, as do the cell types and the biomarkers. The use of standard ontology terms for all the components of the ASCT plus B tables enables scientists to describe anatomical structures, cell types, and biomarkers with precision. People can search the tables using the ontology terms. The use of ontology terms allows them to be confident that they can access all of the relevant data and that they can evaluate all of the information that has been stored. The ASCT plus B tables represent anatomical structures using terms from an ontology for vertebrate anatomy known as the Uber Anatomy Ontology, or Uberon. How do you get to see what terms the Uberon ontology provides? There are several repositories that store information about biomedical ontologies. The most comprehensive is a system known as BioPortal, which has been supported by the NIH to provide access to all publicly available biomedical ontologies in the world, currently several hundred. We can use BioPortal to search for the Uberon ontology and see that Uberon consists of more than 20,000 classes or terms, and that users view Uberon at the BioPortal site nearly 1,000 times every month. BioPortal can show us all the terms in Uberon in a tree browser. If we want to focus on what Uberon has to say about the kidney, we can search for that term, and BioPortal will bring us there. We can see that Uberon has a term for the kidney, and subclasses indicating the left kidney and the right kidney, as well as terms for the progenitors of the adult mammalian kidney that are seen at different stages of embryogenesis. Uberon makes distinctions about anatomy that are different from those made by another ontology of anatomy, the foundational model of anatomy, or the FMA. We can visit the FMA in BioPortal and see that it includes more than 104,000 terms. The FMA is also frequently viewed by BioPortal users. The FMA includes terms for the kidney, as well as the left kidney and the right kidney. The FMA models only adult morphology, so it doesn't list the terms for the embryonic kidney structures. In fact, when we compare the FMA with Uberon, we see some interesting differences. Uberon lists the embryonic structures, whereas the FMA does not. Uberon models the kidney as a kind of cavitated compound organ, whereas the FMA models the kidney as a kind of parenchymatous organ, which is a kind of solid organ. Which ontology is correct? Well, that depends on the assumptions that the ontology's developers have made, and whether those assumptions apply to a user's situation. We can't always tell by inspection what those assumptions may have been, and that's why ontologies can make different or even contradictory statements about the same entity. One ontology might model light as a wave, and another might model light as a particle. Every ontology developer has to make assumptions about the context in which an ontology will be used, and those assumptions often govern what an ontology will say about the world being modeled. Ontologies are thus models that, within a particular context, enable us to talk about the entities and relationships within an application area without ambiguity. But ontologies do not necessarily share the same perspective. One ontology may talk about the adrenal gland, and another may talk about the suprarenal gland, but they are really talking about the same thing. However, sometimes the kidney is represented as cavitated and sometimes as solid, and we have to accept that the context in which an ontology will be applied can affect how we characterize the entities that may be modeled. The major contribution offered by ontologies is that they standardize the way we talk about the world. The gene ontology provides standard terms for describing gene products and their behavior. Uberon and the FMA both standardize terms in anatomy, but they do so in slightly different ways. Both ontologies continue to evolve as we learn more about anatomy, and as the developers make new decisions regarding how to model anatomical structures. Biomedicine has hundreds of ontologies that describe different areas of interest. The availability of so many ontology resources offers a great benefit to biologists and clinicians, although the total number of ontologies can sometimes be overwhelming. Fortunately, resources such as BioPortal can help users to identify the ontologies that are most suitable for a particular task, and applications such as HubMap's ASCT plus B tables can draw on these ontologies to standardize their terms, to ease access to complex information, and to help us all to speak the same language when we interact with these computer systems.